Next session, let me introduce next chair, Dr. Margaret Liu. Margaret, please.
Thank you very much, Sean. And also, I'd really like to thank Dr. Yuan and Dr. Jackson for putting the incredibly excellent framework and all of the information and advances. As you know, we've decided that over this series, what we'd like to do is highlight various technologies and also then continue to provide updates. And so we started with this meeting today to be able to provide insights into the initial vaccines that have entered into the clinic, but also we are looking forward, since we are many of us are fundamental immunologists and vaccinologists, to hearing about the clinical as well as the preclinical and immunogenicity data that help to underscore these vaccines that are in development. So our first session is devoted to nucleic acid vaccines. And in this session, our first speaker is Dr. Kate Broderick, who's Senior Vice President at Inovio, talking about plasmid DNA vaccines. So Dr. Broderick, welcome, and we look forward to your talk.
Thank you very much, Dr. Liu, and thank you to David and Sean and Linda and the rest of the committee for the opportunity to speak today. It's a real honour. Yeah, my name's Kate Broderick. I'm speaking on behalf of Inovio. We are a US-based biotech company specialising in DNA vaccines and immunotherapies. Just to give a little bit of a context, we have 2 parallel phase 3 running for our lead program, which is a therapeutic HPV vaccine. And we just recently released some very exciting data on our glioblastoma program for overall survival at 12 months. But today, obviously, we're speaking about COVID-19 and the progress that Inovio has made to date in this critical area. Next slide, please. So I'll introduce a little bit about DNA medicines, maybe for those not completely familiar. We'll talk about why we believe that this technology is very applicable to emerging infectious diseases. I'll talk a little bit about our experience with our MERS vaccine candidate, which I think stood us in a very strong foundational position to move and pivot very quickly to COVID-19. About the preclinical data that we've generated this far. I won't be able to, unfortunately, talk about any of the phase 1 data, but I'll be able to describe the clinical trial outline and then talk about our future development plans. Next slide. So just a very high-level cartoon, essentially, to start, really to kind of set the scene for DNA vaccines and why we think they have some very key differentiating characteristics which specifically make them very appropriate to our topic of discussion today. First and foremostly is their ability to drive really robust immune responses both in the B and T cell arenas. And we really think that as we understand more about the profiles potentially required for protection against COVID-19, that really both arms of the immune system are going to play really very crucial roles. On the right hand side of the cartoon here are some kind of inherent characteristics about DNA. Of course, there's no anti-vector response, so we can boost very effectively. It's been very established for many years now that DNA plasmids are non-replicating, non-integrating. And also something that we're investigating here internally at Inovio is the ability to really cocktail multiple different DNA vaccines into a single vial. So you can imagine from a patient perspective being immunized once, but in fact against a plethora of different disease targets. Just to give an example, we've, for instance, come up with a multivalent hemorrhagic fever vaccine covering Lassa, Ebola and Marburg. And so that's just a single shot, but you would get vaccinated against those 3 different diseases. On the left-hand side of the cartoon, I think is a very, very important strength of Inovio's DNA vaccines, and that is their stability profile. So our bulk product is designed to be stored for long-term storage at refrigerated 2 to 8, so we do not have a need for long-term frozen storage. But in actuality, our products are stable at room temperature for a year and at 37 degrees or higher for around about 2 or a single month when you get up to 60 degrees. So you can imagine, as we're thinking about deploying vaccines all over the world, logistically, certainly not having that limitation of frozen cold chain is extremely beneficial. I'll touch on this a little bit later on, but certainly DNA can be rapidly and scalably manufactured. And last but not least is its ability to really rapidly design constructs, and obviously that's incredibly pertinent today as we talk about COVID-19. So next slide. So let's call out some of these very specific areas where we think that our technology fits really very nicely into emerging infectious disease puzzle. Well, the speed, and I've kind of touched on that already, but when we talk about our COVID-19 candidate, which we call INO 4800, we actually went from downloading the sequence from the Chinese authorities to having a fully designed DNA plasmid in 3 hours, and that was thanks to the use of our pre-designed computer-based algorithm, and it really pulls on many years of optimization, looking at different aspects of the DNA and protein sequence, and is able to use that algorithm to spit out at the end, as you may think, a fully designed vaccine design. The rapidity of manufacturing. Here in San Diego, where I'm based, we use tabletop bioreactors to very rapidly produce small batches of vaccine for our preclinical work. And then, of course, as we think of GMP scale-up, we go into a specific manufacturing facility. And as Nick touched on too, we're able to harness the established regulatory platforms, and in fact the FDA has agreed that they're so familiar with DNA vaccines that toxicology or biodistribution studies are no longer required for new targets. Our safety profile is something that we also think is a very strong characteristic of our vaccines. We have treated over 2,000 subjects in the clinic with our DNA and our delivery technology, and that amounts to over 6,000 doses of DNA delivered. As I mentioned, we really are able to generate a breadth of immune responses, robust B and T cells, and also, of course, the all-important neutralization antibodies. But a specifically interesting element of our vaccine technology is our ability to generate those immune responses in an elderly population. And that was highlighted by an influenza trial that we ran in partnership with the Canadian government, with our oldest subject actually being 89 years old. We have a very favourable duration of response as well. Our lead product, our therapeutic HPV vaccine, we have immune responses detected 3 years post-immunisation, and we've actually demonstrated protection 12 months post-immunisation in a lethal challenge model. On the efficacy standpoint, we were the first group to demonstrate clinical efficacy with a DNA vaccine combined with our delivery technology, and of course published multiple examples of protection of efficacy from challenge. And again, that all-important stability profile, which we believe will really be very crucial when we think about global vaccine deployment. Next slide, please. So nobody listening today, of course, needs any background data slides on COVID-19, but I included this slide because it's a slide that I've been updating as I've been given talks since the outbreak began, and it really soberingly highlights how over the months the situation has become really more and more dire. Back in March, you can see we were around about 3,500 deaths. By May, that had jumped to almost 300,000 deaths, and today we're looking at just under 500,000 deaths. And I was just recently reading an EPI paper suggesting that here in the US we might be up to potentially 250,000 deaths in the US by October, and by October it's a very, very sobering thought, and certainly something that puts in context the need for us to be working as diligently as we all are at the moment. Next slide, please. So our strategy internally was to kind of set out what our plan was and try and really follow that roadmap through. So we were approaching a dual strategy of emergency use and traditional approval through conducting phase 1 studies, and I'll of course touch on those in a few slides' time. We also generated a preclinical, very strong preclinical data set, which we've subsequently published, and also animal data, animal challenge data, sorry, that I'll talk about in a few slides' time. We want to be a leader in getting to efficacy trials as well, and we're very excited to push that development forward in the coming months. And we've leveraged external funding to support that, and I'll touch on the really fantastic partners that we've developed over the last few months. We, of course, as everybody is in the vaccine development community, expanding our manufacturing capabilities so that by 2021 we can deliver hundreds of millions of doses, which will be necessary as we look at the epidemiology of this diseases. And of course, are also expanding our global partnerships. Next slide. So with grateful thanks to our collaborators and partners who have made this program possible. Definitely the Department of Defence, with whom we're working on the DNA manufacturing side. CEPI, who we partnered, of course, for our MERS and Lassa program, but very early partnered with to support our phase 1 development of INO 4800. The Bill and Melinda Gates Foundation, who we've had a long-term partnership and who have also supported us here for COVID-19. Advaccine, who are running our Chinese phase 1 trial. CISRO and Public Health England, who have been supporting our animal challenge work. The National Chinese Laboratory, who have done some of our neutralization work. IVI, who are running our Korean phase 1 trial. The Wistar Institute, with whom we've had a very, very long and fruitful collaboration, DITRA for device support, and finally the University of Pennsylvania, who have been one of our major phase 1 sites. So thank you so much to all of our partners. Next slide. So we felt that we were in a really strong position when we first realised that this outbreak was becoming extremely serious. And the reason for that was we had a very highly advanced MERS vaccine candidate, INO 4700, which is, as I mentioned, supported by CEPI. That had already been tested in a phase 2 trial, so we felt very confident in our ability to both protect from challenge and also generate really strong immune responses in the clinic for a coronavirus. And we felt that that really helped us pivot towards our COVID-19 vaccine development. And we based the design of our INO 4800 product on the same characteristics that we did for our 47 MERS product, as we knew that to have been very successful both in preclinical and clinical models. Next slide. So just a little bit of background on our MERS development, since it seems so pertinent to the discussion today. On the preclinical side of things, as many of you of course will know, the primate model challenge model is a particularly difficult model, and we were extremely happy to be able to publish that we had generated 100% protection from clinical disease in the primate model after 2 immunisations, and actually 75% protection after a single immunisation, and that data is all published. And I should mention that 100% protection rates in multiple separate independent studies and amounts to just sub 50 separate animals, so a really very rigorous data set there. We also showed robust cellular and humoral responses after a single or 2-dose regime, of course in primates, but also in camels, of course the zoological sink for MERS, and of course mice as well. And it was that preclinical platform that allowed us to launch very rapidly into clinical testing. We ran a phase 1 US-based trial and a phase 2 clinical trial in Korea, and we'll also be initiating an additional phase 2 trial with the support of CEPI in the Middle East and Africa in the coming months. With that candidate vaccine, we showed 76% seroconversion after a single immunisation and 80% seroconversion after 2 immunisations, and displayed strong and broad cellular responses noted at all time points. And as I mentioned on the previous slide, our vaccine candidate was based on the spike protein. Next slide, please. So apologies for this very busy slide, but I do think that it does a very nice job of visually explaining kind of what we, the research team at Inovio, have been doing really since the inception of this project. You can see the spectrum of animal models that we've investigated here. Much of this data is published now in our Nature Communications. And as I mentioned earlier, we've partnered with Public Health England and CISRO to run our challenge studies. And really the culmination of all of this hard work was entering into our phase 1 clinical trial on April 6th. Next slide. So a little bit of the background of how we got there. So as I mentioned, we downloaded the sequence from the Chinese authorities, of which we're incredibly grateful for, on the 10th of January. We ran that sequence through our computer algorithm program. 3 hours later, we had a fully designed candidate vaccine against COVID-19, which we called INO 4800. And of course, in parallel, we did all the standard expression studies to check that it was indeed encoding for the spike protein that had been designed against. Next slide. So this is really a snapshot of some of the data that's published in our Nature Communications manuscript, and that specifically is focusing on our small animal immunogenicity. The large animal immunogenicity detailing the rabbits, the primates and the ferrets will be published in due course. So I can't talk about that today, but I can of course talk about what we've already published. So in mice and guinea pigs, let's start with mice. After a single dose of INO 4800, we were able to really very rapidly see cellular responses at day 7, which continued to rise post second dose. And of course, that's the case also for the humoral responses. We then looked at guinea pigs, and you may ask, why guinea pigs? Well, we were always positioning this vaccine as an intradermally delivered vaccine program, and guinea pigs, it turns out, happen to be one of the best representative models of skin physiology, so it seemed appropriate to do some analysis in that animal model. And you can see here that, as expected, of course, we saw excellent humoral and cellular immunity, but we were also able to assess the ACE2 spike blocking, BAL, IgG biodistribution in the BAL of guinea pigs. Of course, it's vital that the antibodies get where the site of the disease is, and of course also pseudovirus neutralization, and I'll give a little bit more detail on that. So moving on to the next slide. Of course, we also immediately, and I should mention these were all being run in parallel, not sequentially, but you know, just for the sake of the talk, it kind of made sense to do it this way. We of course immediately started looking in the most clinically relevant model, the rhesus macaque. And here's just a snapshot of the data that we have subsequently seen. This is looking at 2 different doses, and this is reflective of what we looked at in the phase 1 trial. And we were able to, again, very rapidly see robust T-cell responses and very, very strong humoral responses as well. And although I'm only showing the humoral responses to the S1 protein, we also looked at S2, full-length spike, and RBD, and showed exactly the same humoral characteristics for that too. So we were very encouraged by this primate data, and in actuality, these animals have subsequently gone on to be challenged. Next slide. So just coming back to those all-important neutralization antibodies, we're very grateful for the collaboration with the Chinese Academy of Medical Sciences, who ran some of our newts for us, and also Public Health England at Portendown, who also have been a fantastic partner. And you can see really bolded out in the green colour for the mice and the guinea pigs, very nice neutralizing antibodies in both animal species. And as we publish our primate data and our ferret data, you'll be able to see the newts that we have derived in those species as well. Next slide. We are running our ferret challenge at Cicero in Australia, and we'll be publishing those data in due course. And we are also running a primate challenge at Public Health England, and also 2 subsequent primate challenges in the US, which really is going to be very nice. That's going to give us 3 independent studies, primate studies, using our vaccine, and we think that will really add to the credibility of our data set. Next slide. So moving, of course, on to clinical development. So we started our phase 1 clinical trial, US-based phase 1 clinical trial on April 6th and fully enrolled on the 27th of April. And we've actually subsequently initiated a phase 1 extension study, and that's looking at patients in an older population and also potential dose sparing opportunities as well. We will be looking to start our efficacy trial in the near future, so keep your ears open for those announcements, and also have partnered with our friends in China and Korea, so Advaccine and IVI respectively, to start clinical development in those countries as well. Next slide. So a little bit more clarity on our US phase 1 approach. This is an open label study design. It was in 40 subjects to, of course, get assessment on safety, tolerability and immunogenicity. Remarkably, we went from bench to bedside in 83 days, which is certainly a record for us internally and something that we are very proud of, and very proud that we were able to contribute to really the working towards a solution for a vaccine here. We found, as I'm sure many or all, I hope, of the developers will say the same thing, that our study sites were extremely motivated, as were the volunteers. I think, you know, the PIs mentioned that we could have enrolled many times over, and there was so much interest in the trial. So far, we have had an extremely benign safety profile, and the trial is progressing as we'd hoped. So unprecedented speed, 83 days from plasmid design to clinical testing, and completing enrolment remarkably in 17 working days. And so those were, just to mention, that was at 2 sites, one at the University of Pennsylvania and the second at AMR Clinical Design site in Kansas City. Next slide. So further clinical development, as I mentioned, you know, we are moving towards announcing our preliminary phase 1 data and having that published as soon as possible for peer review, and then also, of course, launching into efficacy testing as soon as possible. We feel that we've generated now a very strong preclinical data set, and are doing the same for the animal challenge, and that will really support us in conjunction with our clinical testing, enabling a potential EUA. And of course, we're hoping to be a leader in getting into those all-important efficacy trials, hopefully in the July 2020 timeframe. And going to the next slide, which is my last slide. So that just really leaves me to thank you all for listening, thank the team at Inovio for their unwavering dedication, and all our partners and sponsors. We're very grateful. And thank you very much for your time.
Thank you very much, Kate, for giving a thorough presentation. We're running late, so I just want to ask what seems to be the most prevalent question, which is that do you think that since this requires an electroporator device, apparently, although your photo just showed a needle injection, but could you answer a number of people have asked whether or not this will be something that the device can be utilized on a global scale as well for the number of doses?
Yeah, absolutely. And over many years now, we have been developing a very compact device. So I think historically what people think of electroporation devices is something quite bulky, quite sizable. Well, we've really been able to change that paradigm, and now our intradermal delivery device looks essentially similar to an electric toothbrush in size. And we think it's extremely deployable. It's been designed for use in LMIC countries, had extensive usability testing and deployment testing as well. So we think it's perfectly positioned for deployment of a global use vaccine.
Great, thank you so much. I think we need to move on now to our next speaker, who is Dr. Barney Graham. Dr. Graham is the Deputy Director of the Vaccine Research Center at the NIAID at the NIH, and he's also the Chief of the Viral Pathogenesis Laboratory and the Translational Science Corps. And he's done a lot of work on many vaccine clinical trials and areas, but his background in terms of understanding the receptor binding specifically of viruses such as RSV has been, I think, really provided important additional insights for us in terms of designing a coronavirus vaccine. So Dr. Graham, we welcome you.
Yes, thank you, and good morning to everyone, and good evening. Next slide, please. I'm going to go through what we have as a conceptual framework for pandemic preparedness and talk a little bit about the technologies that make vaccine design precision and speed feasible for these kinds of responses. Go through what we've done on MERS coronavirus, and then give you some information about what we've done with the new mRNA for SARS-CoV-2. Next slide. I work at the Vaccine Research Center, which was founded about 20 years ago, and to develop an HIV vaccine. And HIV has driven the technologies that are being used to solve other old problems, and including new emerging infectious diseases. We're privileged to have access not only to the basic science facilities, but also to process development, pilot manufacturing, our own clinical trials unit, and a GLP lab that studies the clinical samples. We've generated and done trials on a number of different vaccine platforms shown at the right, and distributed these products around the world. And the center there, you see the principal investigators and program heads that lead this group. Next slide. So we consider three major approaches or concepts important for pandemic preparedness. One is platform manufacturing, which you just heard about with DNA, which is a plug-and-play type of idea where you can insert your vaccine antigen of choice into your platform of choice and use your well-studied approaches to manufacturing and do rapid manufacturing. CEPI and WHO have both emphasized priority pathogens, certain ones that should be of top concern, and we've tried to advocate for a prototype pathogen approach, which is a systematic approach to all virus families that have the capacity to infect humans. Next slide. These new technologies that I'm listing here have really all emerged to practical use within the last 10 or 12 years, and it's really transformed vaccinology as we know it. I'm going to talk more about structure-based vaccine design, but single-cell sorting and sequencing and bioinformatics have allowed us to rapidly isolate human monoclonal antibodies, define antibody lineages, which can be now a new molecular target or endpoint for clinical trials, and to do single-cell analysis of clinical trial responses. Protein engineers have learned how to make self-assembly nanoparticles, which create orderly displays of antigen that can improve magnitude and breadth of responses. And can you just hit the button one time? Those technologies help us do better precision in our vaccine design and precision in our immune response targets. We also have rapid DNA synthesis, which allows us to rapidly make reagents for vaccine development programs, and recombinant engineering technology allows rapid cell line development, animal model development, and we'll talk more about, as we just did, nucleic acid and vector-based delivery of vaccine antigens, which is platform manufacturing. Next slide. These create speed. And so we think that both precision and speed is important in not only a pandemic response, but in responses to any pathogen of interest. Next slide. So I'm going to start with a few comments about respiratory syncytial virus, which is shown on the F protein is shown on the top left. Parainfluenza viruses also have an F or fusion protein, and they are class 1 fusion proteins analogous to the coronavirus spike. And even though they have different domain structures, they all have the same properties in terms of a fusion machine, and even though they have different shapes, they all cause entry into cells in a similar way. After triggering in the lower panel, the endoproteolytic cleavage reveals a fusion peptide, a hydrophobic fusion peptide that inserts into the host cell. The heptad repeats fold back on each other, causing hemifusion, then full fusion, and you end with a 6-helix bundle that is a non-functional post-fusion molecule. So understanding the function of these class 1 fusion proteins helps you understand mechanisms of neutralization. Next slide. This is demonstrated in RSV, where after a few years, it's triggering by itself, I guess. After a few years, we were able to solve the prefusion structure in the middle, and that revealed a new target that hadn't been known before of high neutralization sensitivity, pictured in red. And this molecule has a tendency to flip spontaneously and be easily triggered. And this post-fusion form of the molecule, you see the red surfaces that antibodies recognize are lost. And as that's been used in multiple clinical trials over the last 40 years, the result is always the same. You can boost neutralizing activity by around 2 to 3-fold, but that's about it. Next slide. But if you can find ways to stabilize the molecule, as we did with a C-terminus trimerization domain fold-on and some internal mutations, disulfide and cavity filling mutations, to maintain the surface contours of the prefusion molecule. Next slide. You can achieve up to 16-fold boost in neutralizing activity, which now makes this a candidate for things like maternal immunization. Next slide. So solving the atomic structure revealed this new target of vulnerability, and we now have clinical proof of concept for structure-based vaccine design and the importance of preserving neutralization-sensitive epitopes on the prefusion structure of class 1 fusion proteins. And we think that concept can be generalized to other virus families. Next slide. We've shown this in a number of paramyxoviruses. I'm just showing briefly here Nipah, measles, and mumps F fusion proteins that have been stabilized and have been shown to have much better immunogenicity for neutralizing activity than the post-fusion counterpart. Next slide. After MERS emerged in 2012 and 13, we also focused on solving structures for prefusion spike molecules, and were first successful with the HKU1 spike shown here, which was inherently more stable than the MERS and SARS, which we had trouble with. And once we had that structure, we were able, in collaboration with Jason McClellan, who was at Dartmouth at the time, Andrew Ward at Scripps, we found stabilizing mutations by putting 2 prolines at the top of the central helix, shown in the upper right panel. And that largely stabilized the molecule. Next slide. And luckily, those same 2 prolines, when you aligned the sequence with MERS and SARS coronavirus spikes, also stabilized those spike proteins well enough to solve those structures and allow us to preserve those neutralization sensitive sites at the apex of the prefusion molecule. More importantly, in the upper right panel C, not only did it stabilize and make the structures more uniform, but it increased expression. So MERS, for instance, expression of the spike trimer increased by 50-fold in transduced cells, which could be an advantage and favorable for gene-based delivery. Next slide. It also allowed us to solve structures for alpha coronaviruses and other endemic beta coronaviruses, veterinary coronaviruses, and even SARS-like bat-derived coronaviruses. Next slide. And because of work we've done with Moderna over the last several years, we started in 2017 work to do demonstration projects for pandemic preparedness, both in Nipah and in MERS coronavirus. And showing you here an example of the MERS coronavirus stabilized spike delivered by an mRNA vaccine in mice, and just showing here that the stabilized version of the spike has dose-sparing effects and has good neutralizing activity preserved down to just a few picograms of product. Next slide. When you achieve those levels of neutralization, in this regimen I have on the upper left of a prime boost at 0 and 3 weeks, and then challenge at 7 weeks, in the panels to the right, you see that 1 microgram and 0.1 microgram doses given twice fully protected lungs from MERS coronavirus replication in this human DPP4 transgenic mouse done in collaboration with Ralph Barrick. Importantly, at the 0.01 microgram dose level, there was breakthrough infection, which you see is that striped bar, and that was associated with some lung hemorrhage, which can be seen grossly on the surface of lungs. And so the question is, does partial immunity result in partial protection, or does it result in enhancement of disease, which has been a concern in coronavirus vaccinology for some time. And you see the green weight loss curve is the group that broke through. weight was lost and then it was regained. And so if that was enhanced disease, you would see that green line on the left side of the gray line. So this is an example of partial immunity giving you partial protection, which is what you would expect if you're not having enhancement of disease. Next slide. So we learned that these proline mutations can stabilize the spike proteins, and the spike proteins that are stabilized are more immunogenic and also express better, and we thought it could be a general solution for beta coronaviruses. But as we went on in this demonstration project, we were taking the MIPA F glycoprotein stabilized, expressed by RNA forward, and through ferret studies and planning to do the clinical trial, phase 1 clinical trial, when next slide, the new coronavirus emerged, and we decided to switch to something that was happening instead of something that might happen. And so we heard rumors, next slide, on about the 6th of January that this could be a beta coronavirus. And as you heard before, when the sequences were released on the night of January 10th, we woke up on the 11th and saw the sequences, ordered constructs with our stabilizing mutations. Next slide. And began communicating with our collaborators at Moderna. And so the first thing that was done was to make the protein from, and we were able to rapidly, with Jason McClellan, who's now at UT Austin, solve the structure of the spike protein. Being able to see this in the right conformation, knowing that it was the right thing to make and the right antigen, gave us a lot of confidence, since we didn't have any monoclonal antibodies yet or even sera to test antigenicity. Next slide. So once we had that protein, next slide, we were able to the bottom left, set up ELISA assays. We were able to use that protein for probes for B cells to isolate new monoclonal antibodies that are now in clinical testing with Lilly and Epsilera, and also apply this new structure to vaccine development. And we are working with Moderna, as I said, on mRNA, but you could also use this protein in other delivery systems, as we'll discuss later today. Next slide. And so I don't have a pointer, but I'll just briefly say that we decided on the specific sequences, amino acid sequences, to use over the next 2 or 3 days. And then Moderna made a decision at risk to start GMP manufacturing before we really knew this would be a pandemic event, because we wanted to do the demonstration project, and they were able to deliver GMP quality material to DMID within 38 days, and to start a clinical trial, phase 1, in about 65 days from the time we got the sequences. Clinical trial was organized and performed at University of Washington and Emory University. And so the main reason to go fast initially is so that we can get vaccine in before the next winter season. So we decided to advance the products, next slide, using this strategy where phase 1 clinical was triggered by immunogenicity data, the phase 2 was triggered by protection in animals, and phase 3 was triggered by safety analysis based on the presence of good neutralizing activity and the right kind of T-cell response pattern. Next slide. So here's data. Some of these data were just posted on bioarchives last week. So this is data in mice. We studied this in 3 different strains of mice. I'm just showing you here BALB-C. On the upper, starting at the upper left, you see either 2 doses or 1 dose of either 0.1 or 1 microgram, eliciting good binding activity. And at 10 micrograms, good binding activity at just a single dose. That's associated with neutralization titers around 1,000 or more for the 1 microgram dose after 2 doses, and up to and over 1,000 in the pseudonude assay for the 10 microgram given once. And that translates on the right side to viral loads that show protection in lung after 2 doses at 1 microgram, and protection in nose in the bottom panel, also all but 1 animal. And about 2 log reduction in the lung for the 0.1 and half log reduction in lung for the 0.01 microgram dose. 1 dose challenges 7 weeks after the single immunization. Both 1 and 10 microgram doses protected the lung. Next slide. So we also were able to show that in mice 13 weeks post-boost with the 2-dose regimen, 1 microgram still protected the lung and also protected the nose. And importantly, since we had breakthrough infection at these lower doses, we were able to look at lung pathology and assess the potential for vaccine enhancement of or aberrant pathology. And in the upper 2 panels, you see PBS groups that on day 2 had some acute lung injury with neutrophilic nodules you see there, and on day 4 had thickened alveolar septa and mononuclear infiltrates that you can appreciate. But the 2 breakthrough doses shown in the next 4 panels at either 0.01 or 0.1 showed relatively little inflammation, and even though they were not protected from virus replication, they did not have these thickened alveolar septa or interstitial infiltrates in the same way that PBS, and they certainly didn't have an enhanced disease pathology characteristic. Next slide. We also did extensive T-cell analysis, and shown here are mice that were immunized either with a inactivated whole virus, this DICOV, or conformationally disrupted spike protein. This was a denatured spike in alum, or the RNA. And what you see on the top right panel is that CD4 T cell by ICS, peptides covering either the S1 or S2 component, showed that in both the DICOV or the conformationally disrupted spike, the CDS, there was significant induction of Th2 type cytokines in the red and orange colors, IL-4, 5, and 13. And but in the mRNA at either 0.1 or 1, you saw virtually only the blue Th1 associated cytokine responses. And the mRNA was the only one on the right now on the upper panel to induce CD8 T cell responses, pretty robust, up to 1% of splenocytes. And then only in the bottom right panel, only the whole inactivated virus vaccine induced responses to the nuclear protein, and also including a significant Th2 responses. Next slide. So the advent of precision vaccinology and rapid platform manufacturing, along with other technologies, has provided solutions for both long-standing problems and emerging infections. This mRNA vaccine we call 1273 is immunogenic, protective, well-tolerated, and has no preclinical safety signals at this time. And as was mentioned earlier by Nick, it's too bad we aren't 2 weeks later, because we could show you the NHP and phase 1 interim reports, which are being prepared now and should be submitted for publication within the next week or 2. The phase 2 has started. We're planning toward a phase 3 start in July, and we think, as was mentioned earlier, that it's only because of prior knowledge of spike structure and this proof of principle for this prototype pathogen approach for pandemic preparedness that we could get off to this rapid start. If this had been viruses from another family, I think it would have taken much more time. Next slide. So I want to thank the people I work with. My lab is pictured there, and I can't point, but Dr. Kia Corbett led our small coronavirus team for the last few years before this event happened, and is now leading our group to in this new development program. The PIs and program heads of the VRC all have been very involved, and John Moscola in particular has been involved in the coronavirus vaccine program. Tracy Ruckworth is listed at the top there, who did the T-cell work, and Caitlin Morabito was the project manager for this. Next slide. I want to mention my long-term collaborator, Jason McClellan, who's in the upper right, who I've worked on this for almost 10 years now, and the people at Moderna on the left, and collaborators at Vanderbilt and UNC, Ralph Barrick in particular, who has done all these mass challenge studies. Next slide. And I'll stop here just illustrating how many groups have helped, both government and non-government groups, to get this started and going. And so I want to thank all of them and for the extraordinary collaborations that have happened during this event. Thank you very much.
Thank you very much, Barney. That was a really nice presentation. And we appreciated, I think, all of us seeing the data on the preclinical results and also understanding the spike issues. So we have a question from the audience about the NIH, and your group in particular has done a lot of work using DNA for other emerging diseases such as West Nile virus and Ebola, and has actually shown some very good, even in older people, which of course is one of the key groups that would need to have a COVID vaccine. You've seen some good immunogenicity with your West Nile virus vaccine, for example, using DNA. So the question was whether or not you would do both DNA and mRNA technology, or did you decide that this was mRNA is the way to go for now?
Well, as you noted, we've done a lot of DNA vaccine work over time, maybe more enrolled people than anybody. And we made a big investment in DNA in 2016 for Zika, and made a DNA vaccine for that. And we can control our own DNA manufacturing. For this, because of our collaboration with Moderna, I like the nucleic acid platform in general, and because of our collaboration pre-existing with Moderna on this pandemic preparedness, we went with the mRNA this time. And I have confidence that their technology can deliver potent responses, whether you're forming particles or not. Our impression with the West Nile and Zika was that if you could make a particle, that DNA could effectively make primary immune responses in the way we deliver it, at least with needle-free injection devices. So we decided on mRNA this time, both because of the commitment and the speed and needing a commercial partner to go through the advanced development phase.
Thank you again very much. And one of the beauties of our format here of having sequential virtual meetings is that we can, in future meetings, be hearing updates on the different technologies that we have at the different meetings. So thank you again. And I'd like to then close this session on nucleic acid vaccines and turn it back to my co-chairs here. Thank you.
Okay, I will introduce the next speaker, next chair for next session. Thank Margaret for doing a great job of controlling time, and also thank all the speakers so far. We are enjoying. We got a lot of good response from everyone, including Gates Foundation sent us an email saying we are doing great job. Okay, so I want to share with the speakers and the audience as well.